In the early to mid-1900s, scientists were noticing an unusual phenomena. Cancer-stricken patients would start to show signs of tumor reduction when they became infected with a virus. At first, this seemed like an anomaly, but it became hard to ignore for a couple of reasons. The first being the frequency of these incidents. They were rare, but did keep happening and were being reported by different doctors. The second reason was that the cancer symptoms subsided with the onset of the viral infection and returned when the virus symptoms went away, meaning that this was no coincidence. Scientists tried to recreate this phenomenon in trials during the 1950s and 60s by injecting live viruses directly into a group of cancer patients, but with little to no success. Fast forward to the 2000s, however, and a blood cancer patient was in full remission after an injection of the measles virus. Another patient, this time with brain cancer, had his tumor completely killed off for a brief period after the cold virus was injected directly into the cancer cells. And finally, a patient with bladder cancer had his tumor completely wiped, thanks to the injection of another strain of the cold virus. So, what's going on here? Why are scientists using viruses as treatments for cancer? But more importantly, how is this actually working? The notion of using a virus as cancer treatment is as fascinating as it is somewhat bizarre. After all, viruses are parasitic. Their interaction with any host is for the benefit of the virus. In fact, viruses will kill the very host it's inside if it isn't dealt with. But at the same time, viruses are unique entities. Their compact and simple structure make them perfect candidates for a multitude of studies and experiments. As a result, they can help us understand very important topics like genetic diseases and cancer. When it comes to combating a tumor, there are three variables in virus-based treatments: the immune system, the virus, and the tumor. The immune system and the virus can be put on one side of this equation, and the tumor can be put on the other. To understand this process, we have to first understand how the immune system in tumors function. Let's start off with the former. You see, part of how your immune system works is by a system of what can be described as markers. When a cell in the body is injected or killed by a virus, it leaves a marker known as an antigen. This acts like fingerprints in a crime scene. The immune system is alerted and will track down the pathogen using this marker. Tumors, on the other hand, are very difficult for the immune system to track because they aren't directly caused by pathogens. As a result, they leave no antigens to distinguish them from regular cells. Instead, tumors are destructive mutations that slowly destroy functional tissues in the human body. So, how does the immune system and a virus kill off a tumor? There are three key processes in any virus that we need to understand. To simplify, we refer to these mechanisms as entry, genetic transfer phase, and exit. Let's start off with the entry process. This happens via cell receptors. These receptors are like the keys to a door. Each key can only open the doors with a matching lock. If the key doesn't match the lock, the door won't open. Because of this, a virus can only infect certain cells. In some cases, only certain species. This is why you can be exposed to a particular virus but not be in any danger of an infection. For example, if a virus that attaches to cells in your brain gets exposed to your lungs, you won't suffer any health issues. But once it gets to your brain, you are at risk of a serious infection. The second key process in a virus is the transfer of genetic information. This is important because this is how the virus uses the infected cells to replace more variants. Now, we will get back to this trait later. But for now, let's move on to the third and final step. That is the exit. This exit process is the final step in a viral infection of a cell. This generally will occur when a virus has replicated inside the cell numerous times and eventually causes the cell to burst. This frees up all the new variants to infect new hosts. So, let's go back to our initial equation and give it an adjustment. You see, it's not the immune system and a virus killing the tumor. It's the immune system killing the virus and the tumor. By infecting the tumor cells with the oncolytic virus, it's like you're setting off an alarm in an area where two crimes are happening simultaneously. These viruses that are used in this manner are called oncolytic viruses. The virus will infect the tumor cells, just like how it would infect healthy cells. This will cause the cancerous cells to die and ultimately produce antigens that alert the immune response. The immune system will then kick in and kill off both the virus and the infected tumor cells. While this plan on a biomolecular level is simple, it's very complicated and difficult for scientists doing the preparation in a lab. Selecting a virus that is suitable for treatment is very crucial. As you might have noticed with the previous examples, 3 different viruses were used: the measles virus and 2 different strains of the cold virus. In each case, the viruses were good candidates based on the receptors they had. But even with that, the viruses did not work for at least half the patients they were given to. Why is that? Well, not everyone's immune system behaves in the same manner. Some people have a more vigilant immune system than others. Because of this, treatments can have different outcomes. That's where the virus design comes into play. You see, we have the ability to modify existing viruses. When doing so, there are a few options that scientists have. One is whether or not they should allow the virus to retain its ability to replicate inside a host cell. Allow the virus to replicate and it becomes more effective. But you run the risk of a serious viral infection. Neuter this ability and you could compromise the treatment itself. Is especially an issue when the virus has to be injected outside the area of the tumor. This means that the variants have to circulate inside the body without detection, all the way to the site of the infection. In that case, there is a balance that needs to be had. Inject too many variants, and you risk either an early immune response or an infection. Inject too little, and not enough will reach the tumor site. Another issue is that non-tumor cells in the body can be receptive to the virus. That will have the virus infect them instead of the tumor cells. Because of this, where you are injecting the virus is very critical. In some patients, there may be no clear route for the virus to take without being intercepted by other cells. So, with all that's being said, let's take a look at some of the current available oncolytic viruses that are in use for treating cancer patients. One major one is called TVEC. TVEC, which is short for Telimagene Laherparepvec, which was approved for use in 2015 and became the first viral therapy to be approved for use globally. The virus was created by editing the herpes virus to have its more harmful characteristics muted. This was done by deleting certain genes. The oncolytic herpes virus is then injected into melanoma cancer patients where it infects the cancer cells and multiplies inside them until they die. This will draw an immune response from the body against the tumor. Overall, up to 88% of patients who used this treatment saw improvements in their condition. In one trial, 61.5% of the patients showed signs of a complete recovery after taking the treatment. Now, with that being said, this is only for melanoma. Most cancers have yet to find a suitable virus as a potential option. The location of the tumor is also a big factor in the success of these treatments. Despite these issues, though, more trials are being conducted, and it is likely that we will see more approved oncolytic drugs going forward. Keep in mind, there are many institutions and trials going on and testing these new treatments. But for now, we had to condense to keep this video short. So what do you guys think? Do you think we'll cure cancer this way in the future? Let us know your thoughts. And if you like this video, please make sure to click like and subscribe.